Today I will be discussing the second part of the synaptic transmission. In this, I have two important topics to cover. One, the vesicular transport mechanism, and a brief about the neurotransmitters. Now I have made here the vesicle associated membrane proteins here. I would discuss about this diagram as I proceed. In the last class, what we did, we just covered all things, definition of a synapse and all the things related with the synaptic parts, the functions, the presynaptic parts, so on. So that was the, and even we mentioned about the events happening during synaptic transmission. Today I will be covering these two important aspects, the vesicular transport mechanism at the presynaptic level and about the neurotransmitters. The remaining part, especially the properties of synapse, I thought it is a big topic, and unnecessarily I would be rushing you all, wherein this is one of those important topics. I want to cover it in a separate class that's in the next class. This is what we left in the last class, and here I have after the last class I have added additional point here. So I just arrive here. These are the events: the arrival of impulse, the opening of calcium channel, the calcium channel enters inside, and that triggers the vesicle associated membrane proteins, and then the vesicle moves, come to the active site, gets itself docked, primed, sensitized, and then it is released. That is item number 6. Then the transmitter thus released in the synaptic cleft, this is synaptic cleft, gets attached to the neurotransmitter receptor, and the attachment of the neurotransmitter receptor brings about the ion changes in the ion channels, or opening of the ion channels. The opening of the ion channels leads to either depolarization or hyperpolarization. Depolarization is known as a EPSP, excitatory postsynaptic potential, or hyperpolarization is IPSP. And then once it reaches that particular critical level, that fires an action potential. This is an action potential. It is a postsynaptic action potential, and this brings about the action in the postsynaptic neuron. So these are the various events during synaptic transmission. Just to revise, and I have listed them. I have not gone according to the list I had mentioned, but I had just narrated the events happening. In this lecture, what we will see, how the vesicles move, because if you are looking back here, the arrival of a calcium, the calcium triggers certain things here in the vesicle, and this vesicle reaches to this site here. This is an active site or active region. So how this moves from one part of the presynaptic terminal to the real active zone of the presynaptic terminal. So in that way, if you are looking at, I start from here. So the this is the position of the vesicle right now. I will come back in the entire cycle, and arrival of the impulse opens up the calcium channel, and that calcium is utilized or triggers the membrane proteins here, and these membrane proteins try to attract each other and put them on the tracts. These tracts are those the cytoskeletal structures on which these the vesicles, they are like a containers of, you see that the containers of the ship are stored in a rail, these things just like that, that they are placed on that. That is what we are calling it as a docking. So now they are placed because of the movement of these vesicle associated membrane proteins. They put them or docks them on the particular track. And now this track, utilizing energy, it's getting primed. Again, it is all because of the calcium activating this ATP mechanism, calcium activating these things. They get primed. Now, with this further, the calcium arrives because of the opening of the voltage gated calcium channels. And this calcium further moves the vesicle associated membrane proteins, and that comes, that means these two ends they oppose, and then they come here, they release the contents of this sac. This is a sac there. This release the contents of this sac, that we call it as a exocytosis. So that means there are two processes happening here. One, the fusion of these two membranes, and then the release of the contents of this sac outside. And this release is not, it's an explosive release. It's not a very simple release as far as the synaptic cleft is concerned. This explosive event releases all or empties the contents of this sac. These contents of this sac is the neurotransmitters. Once the contents are emptied, that is exocytosis, what we call. So then there will be the another mechanism operates, that the clathrin are the coatings. These again another protein molecule, and that makes this thing because they want to recycle the entire this molecule or particular thing, the sac. This the clathrins coat it, and after coating with the clathrins, they will begin. This is fused. Now this gets fused. Whatever is broken, it gets fused, and then new vesicle is formed. This is a endosome. Now this endosome is now having certain ions, especially the H ions and other ions there, and they will come and fuse with the endosome of the entire Golgi complex. So from the Golgi complex, they will be again primed, processed, and what all the, maybe you think like a workshop in which the denting, painting, and everything is done, and then from there again a new bud will be formed, and the new bud, the new pinching of the vesicle or the sac is taking place. And now once the new pinching takes place, the neurotransmitters are pushed into this sac, or put into this sac, and now this sac again comes back to this thing. So that means the whole this vesicle, what I say as a sac, is recycled in the entire period. And in the vesicle recycling mechanism, these parts are happening as from this translocation to this translocation. It's a normal cellular event or a normal event happening in the presynaptic terminal. Whereas this docking, priming, fusion, exocytosis, and endocytosis, these four events, 1, 2, 3, 4, and even fifth event, these are all triggered by the arrival of impulse, and that arrival of the impulse releasing the calcium, trying to move this sac in space in the active zone, and then brings about the entire neurotransmission. So now this neurotransmission, if you are looking at the, say for example, if I hit at the, if you know the knee jerk, what we say as a knee jerk response. Knee jerk response is one of those synaptic event wherein I tap the tendon here, tendon of the quadriceps femoris or the patellar tendon, and then that stretches certain receptors in the muscle, and this muscle, they reach the spinal cord, and then these neurons have a synaptic contact with the alpha motor neuron supplying the quadriceps muscle. At this synapse, they come and activate, they release these things, and once they release, they will activate the alpha motor neuron. So this event definitely will take time. So that means you can think about the time taken. If it is too long, you will not be able to attend. Say, for example, if you are touching a hot plate, if you do not withdraw at a appropriate time, you will burn your hand. So that means it's what is called a reaction time. It's part of the reaction time here. So all these processes. Besides, there is something happening here in the synaptic cleft and the postsynaptic event. That's a different time interval altogether. Now with this thing, I want to go little further. So now how that thing happens, what I talked about the vesicle. So this moves the vesicle associated membrane proteins. What are those vesicle associated membrane proteins? Where are they and how are they? If I have just made here a diagram to substantiate for this diagram, I have taken it from Genong. I am trying to put that thing in place according to this diagram. So this is the presynaptic terminal. This is the membrane of the presynaptic terminal. You can just see that. And in this presynaptic terminal, this is the vesicle. This is the boundary of the vesicle. You can just see that. And it has two proteins attached, one protein here and one protein here. This is a protein which is attached to the vesicle. This is known as a synaptobrevin. They are also known as a snare proteins. Snare proteins, since it is attached to the vesicle, it is a V snare. V snare. This is a V snare. And we have another molecule or another protein here. This protein is attached to the membrane. This is the membrane there, this membrane. So you can just see that this membrane, this is syntaxin. This syntaxin is attached with this membrane, and this is known as a T snare. So that means one is attached to the vesicle, one is attached to the membrane. And now these two are attached, but they are two separate proteins. Now for them, some intermediary proteins like synapse, we have the synapse here and we have NSF there, and this RAB, the MUNS, and the SNAP25, especially SNAP25 plays a role because SNAP25 is attached to the syntaxin, and then SNAP25 tries to fold and try to bring in the vesicle. And the RAB protein trying to move the vesicle towards the membrane, and this trying to attract it. So now these are all the proteins, and that is how they are. So just to list, we have a synaptobrevin, we have a syntaxin, we have a SNAP protein, SNAP25, and we have a alpha and gamma synapse, and we have NSF, and these are some other type of proteins which are associated with this membrane transport. Now, to go a little bit, so this event, what we are trying to think about, the movement from the vesicle coming to the membrane and getting it ruptured. So these three things are very, very quick and explosive events. If you are coming back here, so these are the proteins. You can just see this blue one is the synaptobrevin. The blue filamentous structure is the synaptobrevin. This is the synaptic vesicle. This is synaptic vesicle. Now, in the first event, wherein we are here, so that means it is not attached to this thing. Maybe you can go back here, somewhere here, or this one is moved and putting into the dock. So that means we are trying to come this side. So now once you are here, so then it gets attached. So now we have another protein. This is the green one. Green one is a SNAP protein, the SNAP protein. Then this red one is a syntaxin. So the color codes, you can just see the color codes there. So the syntaxin is the red one. The green one is the SNAP25, and the blue one is the synaptobrevin. This is how the arrangement. If I go back to this thing, the synaptobrevin there, syntaxin there, and the SNAP25. That means these are the three major proteins for which the movement of the vesicle to the membrane, towards membrane is taking place. Now this is the first event, and because of the release of calcium, now what happens? It activates this SNAP protein. Now what this SNAP protein does, you can just see that this is the configuration of the syntaxin. This is syntaxin there. This is the configuration of syntaxin. This is loose. This is loose there. This is loose there, and it is not a relaxed state. So now this one is getting folded. So you can just see that there is a folding of this SNAP25. And now this folding of this SNAP25 pushes the, because if you just see the arrangement, it's a sort of a trying to be hypothetical or whatever. But if this protein, this folding here, this and this thing, or in one this clip, in this clip, you can just see this is a clip, and now this clip is just putting them together. Then what happens? This vesicle which is here, so now that is getting zipped. So this is zippering mechanism. So that means the arrival of the impulse, release of the calcium begins the zippering mechanisms, or zips the mechanism. So that means it tries to put them together. Once it tries to put them together, so that means it moves this thing because of the folding. That means both of these proteins, they are pulled. So this is the holding protein to the membrane. The red one is the holding protein to the membrane. Blue one is the holding protein to the synaptic vesicle. So now they are pulled. That means synaptic vesicle is pulled, because now you cannot pull the membrane up. Membrane is a larger structure than the synaptic vesicle. Now the synaptic vesicle moves along with this thing. This is the docking system. This is the docking system. So once the docking system is happening, so now what happens? There are certain inherent properties. Once you have these things, the inherent properties, so that means the membrane gets fused. And this is called a constitutive properties. So that means it activates certain systems so that the energy is released. So now it gets fused. It gets fused with the membrane. This is a plasma membrane here. It gets fused with the plasma membrane by the inherent properties or by regulated properties. What is that regulated? Regulated is again because of the calcium. Now calcium activates certain enzyme systems which tries to move the, or which tries to make the attachment of this or fusion of this membrane, because again all the things are covered by the unit membrane. They get fused and they become one. So both of them pull them together. So now this is a very what I say, the explosive process. So that means even if this fails, it regulates so that it is pulled, and then it opens here, it ruptures, and it will release the contents of this. This is the release of the contents. So these contents are nothing but neurotransmitters. So once they release, immediately it will be unzippered. It is zippered here, or it is zipped here, and now it is unzipped. So once unzipped, it will be the whatever the synaptic vesicle is there. The synaptic vesicle is getting emptied and put back forth. So that means this is what is told as a kiss and run. So kiss is this is kissing the membrane there, and once the kissing, explosive release of the contents of the vesicle, and then running away, and again emptying and running away. So this is a exocytosis. So I have mentioned here 1, 2, 3, 4, 5. One is attachment. Attachment with these things are getting triggered or priming. What we call it as a priming, and then comes a zippering. So this is the second event. Then the exocytosis. This exocytosis is brought about by the inherent or constitutive mechanism and the regulated mechanisms. They release the contents, then that means this is setback, and that will go again translocated and fuse with the endosome, and then again a new vesicle is formed, and that vesicle is again added up with these things. So that cycle continues. That is the entire presynaptic event. Now, what is the important? What is important about this? Now, why we are worried about this particular event? Here I have mentioned here BONT, botulinum toxin, neurotoxin, botulinum toxin. TENT is a tetanus toxin. You know, tetanus, you must have all must have been heard about it. You take the tetvac. This tetvac is the vaccination against the spore-bearing organism, that's anaerobic organism, tetanus. This tetanus, produces a toxin, and that toxin is very lethal. It produces convulsions in human being. Similarly, botulinum toxin, it is the much great potent toxin than the tetanus toxin. Again, it is also a botulinum toxide, is again another group of spore-bearing bacteria or anaerobic bacteria. So now what happens? If you are looking at BONT, now there are a number of forms of the BONT. BONT-G, BONT-B, BONT-D, BONT-F. All of these things inhibit synaptobrevin. This blue one is the synaptobrevin. So now they inhibit, they will not allow it to activate. So that means they will not allow this synaptic vesicle to reach and come to the site and get it released. So that means that prevents the release of the toxin. Okay, so now if you are looking at the TENT here, TENT is happening here. The sites used for TENT and BONT-B are same. So that means tetanus toxin and the botulinum toxin B are in the same level. Now, so you have another the format isoform of the botulinum toxin C that will be inhibiting the syntaxin, the green molecule there. That is which is attached to the membrane. That is what is a V snare. So then we have the BONT C, A, and E. So these are attached to the SNAP25. SNAP25 is the one which tries to fold or put them in the clip, these two proteins. So this is SNAP25. So that means you can see that the botulinum toxin is acting at a multiple form, one at the synaptobrevin, one at the syntaxin, one isoform at the syntaxin level, one isoform or some isoforms at the SNAP25 level. And thus they prevent the release of a transmitter from this into this, whatever may be the thing, whether it is a tetanus toxin or botulinum toxin at 3 different levels, tetanus toxin at only 1 level. That will prevent the release of this thing. So now I'm just trying to put that thing in words here. These toxins, especially the botulinum toxin and the tetanus toxins, they block the neurotransmitters or prevent the neurotransmitter release from the vesicles. They are endopeptidases that cleave or inactivate the proteins. What are those proteins? Synaptobrevin or syntaxin or SNAP25. And once they cleave that or inactivate that protein, the fusion or exocytosis will not be possible. Thus, they prevent the fusion and exocytosis of the vesicle. TET toxin, that means tetanus toxin, or Botox, B, D, F, and G. What I have shown in the B, D, F, and G, or acting at the synaptobrevin level, which I mentioned. Botox C is inhibiting or acting on the syntaxin level. Botox C. You can just go back here, Botox C here. This is Botox C. So now, then Botox A and B, Botox A at SNAP 25. You can just see that this is Botox A, C, and E. So they are at a SNAP 25. Now, so that means main purpose is the difference. So if we are looking at the difference in the botulinum toxin and the tetanus toxin, so it's the tetanus toxin is only with the synaptobravin. Botulinum toxin is with the synaptobravin, syntaxin, and a SNAP 25. That means it's more complex or more potent. Clinically, tetanus toxin causes what it does. It does the it prevents the release of the presynaptic neurotransmitter, and the release of the presynaptic neurotransmitter that produces the that means there is what is called a presynaptic inhibition. That presynaptic inhibition does not happen. Presynaptic inhibition does not happen. Usually what happens, there is what is called a recurrent inhibition. So that means once the motor neuron is coming from the alpha motor neuron and its collateral goes back into the spinal cord, and through another Golgi bottle neuron, what's called a Ransa introneuron, that interacts. So now for the Ransa introneuron, this gives a synaptic contact. So now this synaptic contact is inhibited. That means the presynaptic inhibition, that means the release of a transmitter is inhibited. Thus alpha motor neuron is not inhibited. It is inhibited presynaptically. The release of transmitter is inhibited. Thus the alpha motor neuron is not inhibited. If the alpha motor neuron is not inhibited, you will get the excessive activity of the muscles, that is convulsions. That is one of those things we see with the tetanus. So now the botulinum toxin, if you are thinking about a spastic paralysis and a placid paralysis is a total paralysis. The spastic paralysis is because of the activity, some of the gamma motor neurons are involved. So that means that there is a spasticity. So this is a placid, means become flat. So this flat paralysis is by blocking the acetylcholine either at the central level in the spinal cord and at the neuromuscular junction. In both the places it happens. So that means botulinum toxin also blocks the neuromuscular junction. So now, so what? Okay, that's what we have understood. That's what we are worried about. That is why we are taking Tetvac whenever we make the street accidents, whenever we feel that there is dirt and dust which we are exposed. Now, if you come back, this monster botulinum toxin now has been put into use. That means put into a better use or a medical use. How? Can you think about it? So you have a disease known as achalasia cardia. Achalasia cardia is a condition where esophagus joins with the stomach. So this esophagogastric junction is guarded by a physiological sphincter. And this physiological sphincter is having the acetylcholine receptors. In this condition, this acetylcholine receptors or acetylcholine transmission is excessive, so that there will be a spasm of this sphincter. So this will not allow any entry of food material through the esophagus into the stomach. Now, to help these individuals, to help the achalasia cardia patients, what we do is the medically a small dose of Botox is injected. So what it does, it decreases the hyperactivity induced by the cholinergic activation here. So once the hyperactivity is released, the person feels comfort. Okay, the one important aspect of this thing, this is not a permanent solution. That means it's a temporary and it may recur after some time again. Okay, that's one aspect. So now you know the most of the Hollywood, the Bollywood, or the even Telugu cine stars, they look always smart. They are evergreen. How they can be evergreen? How they can be evergreen? Especially some of those heroines, they look as though they are very young. Now, how that could be possible? Because they will undergo certain treatment, and they will use this particular toxin, especially the Botox treatment that paralyzes those wrinkles are nothing but the contraction of the muscles, and that would release those wrinkles, and they will look young and fresh. That is the plastic surgery or a plastic component associated. So that means the injection into the facial muscles to remove the wrinkles in certain individuals. Okay, so this is only the aesthetic value, or a value in terms of the plastic for the plastic surgeon and his especially those particular celebrities associated. So this is about Botox. I mentioned about that, and it may be a question. Synaptic, the difference between botulinum toxin and tetanus toxin, and the uses of Botox. I have just mentioned about the uses here. Now, going back, what's the neurotransmitter? Going into further, the neurotransmitter. Neurotransmitter is a chemical, and this chemical may be a simple molecule to a complex molecule. Simple molecule like an amino acid, or even it may be a gas, or it may be more complex like the peptides. So this molecule, what it does, it is released by the synaptic vesicle in the synaptic cleft. And once it releases, that means the electrical energy which is coming from the presynaptic terminal now has been transformed into a chemical energy. This electrical energy has been transformed into a chemical energy. Now this chemical energy has to be converted or transduced to electrical energy again in the postsynaptic nerve. So now this is what the neurotransmitter does. What are the minimum requirements for a neurotransmitter? So we cannot say all the chemicals neurotransmitters. So now to say that the neurotransmitter, to say a particular compound or a chemical is a neurotransmitter, it should be available at the synaptic cleft. Point number one. And it should have all the enzymes or machinery for the synthesis of that chemical, storage of that chemical, and placement of those chemicals in the synaptic vesicles. That machinery should be available, and this chemical should not undergo anything, or it should not interact with the membrane. It gets released into the synaptic cleft upon the arrival of the impulse as demanded or as required. These are some of the presynaptic requirement of a neurotransmitter. Then next, what happens? Now this chemical should have its own identifying recognition sites on the postsynaptic membrane. These recognition sites are known as receptors. These are pharmacological receptors or chemical receptors. So now they should have a specific receptor. So that means we have acetylcholine. The acetylcholine on the postsynaptic membrane, there should be acetylcholine receptors. So now this acetylcholine receptor, that means acetylcholine site should be there and the receptor should be there. It is something like a lock and key. So that would come and sit on the membrane on the postsynaptic cell. Then the interaction of the neurotransmitter and the receptor should lead to either changes in the postsynaptic membrane, electrical changes in the postsynaptic membrane, known as a excitatory postsynaptic potential or inhibitory postsynaptic potential. In the other simple words, either the interaction The neurotransmitter and receptor interaction either should produce the depolarization wave or hyperpolarization wave involving the ion channels. That is the another important requirement for a neurotransmitter. That means if you inject these neurotransmitter iontophoretically on the postsynaptic site, they would produce these. Then comes the, it's not enough. The neurotransmitter should, the action of the neurotransmitter should stop. There should be a mechanism to halt the action of the neurotransmitter. So now there should be a machinery for the removal or halting of the neurotransmitter action. So this happens by diffusion into the synaptic space or synaptic cleft, and its metabolism here, if you are if I'm talking about acetylcholine esterase. In esterase, it's a acetylcholine esterase is an enzyme which splits the acetylcholine into acetate and choline. These two components are recycled again. That's another event that the recycling of the using the materials as a raw material. So that means we should have a mechanism. That means one by diffusion, one by metabolism, as I mentioned about an enzyme system which destroys it, and most of the neurons, most of the neurotransmitters excepting the acetylcholine are taken up by the presynaptic terminal. That is a reuptake mechanism. They take it back. Now for the reuptake mechanism, there should be transporters in the presynaptic terminal. That is a requirement for a neurotransmitter. Say for example norepinephrine is taken up back into the system, and we have glutamate. So glutamate is taken up, dopamine is taken up into the presynaptic terminal, serotonin is taken up. So now these especially the serotonin uptake inhibitors are used to enhance the serotonergic synaptic transmission in certain cases. So that means there should be a machinery for the removal or halting the neurotransmitter action by either by whatever the diffusion is naturally happening, metabolism, and the reuptake mechanism. Now, okay, it's not the only neurotransmitters which are working alone. There are certain other molecules or chemicals which help the neurotransmitters to perform better. These are called neuromodulators. So these neuromodulators are again released from the presynaptic site, and they have a little action, synaptic action. That means they will not produce any depolarization. The only thing, they enhance the activity of the neurotransmitter. So that means they have a very little or a no direct effects on their own, but they modify. Rather, modify is a very tricky word. Modify means either enhance or decrease. The modify or inhibit or suppress, whatever. Alter is it happens on both sides, plus minus both sides. So they will modify the effects of a neurotransmitter as required. These are called neuromodulators. So then about other aspects of all the processes at a presynaptic and a postsynaptic events are regulated by many physiologic factors and can be altered by drugs. So that means either a release of a transmitter or the event happening at the receptor, that means a receptor interaction, or the ion channel blockage. All these things could be altered by drugs. Say for example, even if there is a release of a neurotransmitter receptors, if I knock down the receptors or if I block the receptor with some other antagonist, so then action will not take place. Or if I see that the neurotransmitter sits on the receptor and it produces the action, I will see that ion channels do not open, so I block the ion channels like that. So they can be more altered by a number of drugs or chemicals. So this is a challenge for our medical profession. Thus, one can study the somatic and the visceral activity, but and also the emotion. What is emotion? Emotion is a complex mixture of a synaptic transmission. And if I am angry, there are several synaptic things. They get excited and release, ultimately release the adrenaline and noradrenaline, and even brings about certain hormonal changes. There's emotions. Maybe whether it, the emotion may be a positive emotion or a negative emotion. That means you get depressed. That's also an emotion. Or you get joy. It's an emotion. You get angry. That's an emotion. So all these emotions are associated with the neurotransmitter release. I will talk about those things when I talk about the cingulate gyrus and the hippocampus and other limbic system, limbic complexes. The behavior that means withdrawing or going forward or avoiding, all those behaviors and all other our complex functions. So we perform so many functions. All these things are modulated, and these modulations can be by the drugs, especially in terms of the emotion and behavior. We can have these drugs designed and formulated, especially these drugs are designed and formulated especially for Parkinsonism. If you are looking at, wherein it is a failure of the synaptic transmission, because certain dopaminergic neurons in certain areas in the basal ganglia are not there. So now we may have to enhance the dopaminergic transmission in the Parkinsonism. Or if you are looking at the Alzheimer's disease, what is happening? The cholinergic neurons in the hippocampus and around the circle of main heart, they are destroyed. And because of that, the person is forgetting. He does not have the short-term memory. So now you have to enhance the uh, the the synaptic activity of the cholinergic neurons. So like that, we can use these drugs to enhance these various events about the neurotransmitters. Now coming back, what are the various major various neurotransmitters? If I directly ask you a question, what if I were taking a class, I would have heard only acetylcholine. Everybody would have said acetylcholine. Yes, agreed. Agreed. Acetylcholine is a neurotransmitter. But acetylcholine, the more than acetylcholine, glutamate is the major neurotransmitter in the entire nervous system. I have written the glutamate brother or sister, whatever I can say, sibling, aspartate. These are excitatory amino acids. Glutamate, aspartate, kainate, quisquelate, and all those things. I have not written intentionally. I have not written the kainate, quisquelate. So glutamate is the major neurotransmitter of the central nervous system. It is an excitatory neurotransmitter. So acetylcholine, I have just mentioned here. These are the receptors, the nicotine and muscarinic receptors. It brings about the action. Then glutamate, nicotine receptor is a ion channel receptor. Muscarinic receptor is a ligand-gated receptor. So that means now I want to say that the ion channel receptors and ligand-gated. So that means ion channel receptors means sitting of the acetylcholine, it opens up the cation channels of the nicotine. So that is ion channel. In case of muscarinic receptors, here it's the ligand, the muscarinic acetylcholine sits on the muscarinic site. So muscarine is a alkaloid from a mushroom, and this sits on the ligand. This ligand is specific for, or it is highly potent for that. So that means that is why it is muscarinic. And these muscarinic receptors, upon the acetylcholine sitting, brings about certain changes. It may be adenyl cyclase or cyclic AMP or cyclic AMP mechanisms that in turn modulate the cation channels on the membrane. So that means there are signaling pathways in these things. So these are, one is a ion channel, then the receptor is ion channel. This is a ligand gated ion channel. So that is one. Glutamate also, we have two things there. So this is again similar to nicotine. It opens up. This is a voltage dependent. The AMPA site that opens up the channels and the cation channels, and that produces a depolarization. Whereas the NMDA is again a ligand-gated ion channel, ligand-gated channel. Serotonin, again, we have in the serotonin, there are multiple receptors. The 5HT1 to 5HT7 and 5HT1 A, B, C, 5HT2 A, B, C, D, like that, 5HT3, 5HT4. So many are there. So these overall, this family may be around 20 receptors are there, or more than 20 5HT different class of receptors are there. I'm not going to bother you. It is the pharmacology or the research. If you take on, you will get it. But these receptors, some are ion channel receptors, some are metabotropic receptors. Now I have used another word, metabotropic. Here I have this opens up the ion channel. This sits on the ligand-gated channel. And the third is the metabotropic. Metabotropic receptors are those receptors. So that means we have in the glutamate, we have the metabotropic receptors. So that means it activates. Again, it is something like a G protein coupled receptor, and that sits on that, and that brings about the nuclear changes. That means what are those nuclear changes? These nuclear changes are either a transcription or a translation, which brings about the long-term effects. Long-term effects, be synaptogenesis or dendritic spine formation or a synaptic plasticity, and all those things are the metabotropic, or that metabo means metabolic changes. Metabolic changes happening in the synaptic site. That is the another receptors I was talking about. Histamine has 2 here, H1 has 2. Dopamine has 2. I have just maybe there may be in the recent books D2, D3, something like that. I'm not bothered. The mostly available I'm just mentioning. The glycine has glycine A and B. So I have written type 1, type 2 here. Glycine A, glycine B receptor. And norepinephrine, we have mostly it is alpha receptors. Sometimes very high concentration, it may also activate the beta adrenoceptors. Epinephrine, it is mostly, it is both equal on both sides, alpha and beta adrenoceptors. GABA has 2 receptors, GABA A and GABA B, and GABA A opens up the ion channel, chloride ion channel. GABA B is a sort of a metabotropic or sort of a ligand-gated receptors, which opens up the potassium channel, or which allows the potassium, or which allows the potassium channel. So substance P, the enkephalins, endorphins, these are called endogenous opiates, the neurotensions, and several peptides like somatostatin, thyrotropin releasing hormone, gonadotropin releasing hormone, VIP, arginine vasopressin, that is ADH, angiotensin 2. So many, you can have a very vast list, maybe 150 to 200 peptides there. They have some amount of a neurotransmitter activity. Okay, so besides these major neurotransmitters, there are 3 gaseous neurotransmitters. So what are these gaseous neurotransmitters? One is the nitric oxide. Then second is the H2S, that is a hydrogen sulfide. So we know that we are worried about in chemistry practicals. It is awfully smelling whenever I have been as a student of Intrascience or pre-university. We try to be worried about performing the experiments with H2S. It is awfully smelling. Then we have a carbon monoxide. These 3 gases are neurotransmitters. Now, once I have listed the neurotransmitters. So now I should be able to tell at least without thinking 5 of them at any given point of time, starting with the glutamate, acetylcholine, serotonin, histamine, dopamine, glycine, GABA, substance P, neurotensin, angiotensin 2, so on and so forth. You can just say that. Now, I have just categorized here as excitatory transmitters and inhibitory transmitters. So now, acetylcholine, It may be excitatory nicotine and muscarinic receptors, but muscarinic receptors at the heart is inhibitory. At the heart is inhibitory. If you are looking at the intestine, it is excitatory. So that means again M2, M1, M2, M3 are coming up. The muscarinic 1 receptor, muscarinic 2 receptors, 3 receptors. Then glutamate, we have a glutamate and AMPA and NMDA. I have said again NMDA 1, 2, something like that. I'm not bothered. It's an excitatory transmitter. Serotonin, most of them are excitatory, some of them are inhibitory. So histamine, H1 and H2, they are excitatory. Dopamine, D2 receptors are inhibitory. D1 receptors are excitatory. That means that produces depolarization. This produces hyperpolarization of certain membranes. Glycine B, I said glycine 1 and glycine 2, I said no. Glycine 2 is equal to glycine B. Glycine B is excitatory and glycine A is inhibitory. Especially these glycine A receptors are present in the spinal cord. Norepinephrine, alpha and beta. So like that, but coming back to GABA, GABA A is a chloride ionophore. Chloride ionophore. It opens up the chloride ion channels. Thus, by opening up the chloride ion channels, it hyperpolarizes the membrane. More chloride enters in, and it hyperpolarizes and thus inhibits. So whereas GABA B, it opens up the potassium channels and the potassium moves out, and once potassium moves out, the positivity decreases. That is why the negativity or hyperpolarization. So these two are inhibitory. Then substance P, we have the opiates, the enkephalins and endorphins. These are inhibitory in nature. They will check. So now we have endothelins. Endothelins, we have this ET1 is excitatory, ET2 is inhibitory, and all these things are neurotensin and several peptides are excitatory. The gas is nitric oxide; these are excitatory, and nitric oxide it's something like a double-edged sword. It has an inhibitory action also. Overall, excitatory transmitters are plenty. The inhibitory transmitters are less. Of the inhibitory transmitters, I would focus on first GABA, gamma-aminobutyric acid. That is number one. If somebody asks me a question, what would be the inhibitory? Name the inhibitory transmitter. I would say first the GABA. Then comes these enkephalins and endorphins. Then we have these. Because these muscarinic receptors here also, they are more complex. Muscarinic receptors in the heart is inhibitory. Muscarinic receptor at certain areas in the brain are inhibitory. So that you have to have a specific these things. 5-HT1A receptors are inhibitory. D2 receptors are inhibitory. So it is more complex, and you try to weigh when you are answering. But you never fail if you answer GABA. Okay, so now I have just listed. I will again tell the inhibitory transmitters are GABA first, enkephalins or endorphins, number 2, ET2 number 3, then comes glycine A receptors. That means glycine is a double-edged sword, the glycine. So these are only undoubtedly these two are inhibitory. Now coming back, so now these are some of the neurotransmitters I have just listed. I have taken this table from the Genome. Monoamines, acetylcholine and serotonin, and these monoamines, the acetylcholine and serotonin, they share the space in the presynaptic terminal with these transmitters at some place or the other. That means enkephalin, CGRP, ghrelin. Ghrelin is another inhibitory transmitter. GnRH, neurotensin, somatostatin, substance P, and VIP. Serotonin shares with the CCK, cholecystokinin, enkephalin, neuropeptide Y, and substance P and VIP. Dopamine with these CCK, enkephalin, neurotensin. Norepinephrine shares with the, because why these different neuropeptides are there. So these neuropeptides, they behave like neuromodulators. precisely, they will enhance the activity of these primary. These are primary neurotransmitters, and these are transmitters, but they will be effective in the presence of these neurotransmitters. So I have listed them. You can when I give these things, you can just see they have particular roles to play. So that means glutamate action is enhanced with the substance P, TRH. So glycine, neurotensins, the GABA, the cholecystokinin, enkephalin, somatostatin, substance P, and TRH. So like that, these are some of the neuropeptides which are present. They are also neuromodulators, but they are also neurotransmitters. Now, how the neurotransmitter brings about the action? That's the question. That means intracellular signaling mechanisms. The neurotransmitter, I have already mentioned, the neurotransmitter and receptor interaction directly opens the ion channel, as happens in case of nicotinic acetylcholine receptors. That opens up the ion channels, the cation channel, the sodium moves in, the depolarization, and then produces the excitatory postsynaptic potential and then exit potential. That's one. Same thing with the glutamate AMPA receptors that opens up the ion channel. Whereas there would be ligand-gated ion channels. Ligand-gated ion channels. These ligand-gated ion channels, they sit on the receptor. They are a particular identifiable spot, and these brings about. They trigger this adenyl cyclase cyclic AMP mechanism, or guanylyl cyclase cyclic GMP mechanism, or the guanylyl cyclase or adenyl cyclase inositol triphosphate calcium dependent mechanism, inositol triphosphate and calcium dependent mechanism, or inositol triphosphate calcium calmodulin mechanism, or diacyl glycerol. So maybe you, by this time you are aware of these intracellular signaling pathways. Maybe I would just narrate the broadly the intracellular signaling mechanisms are brought about by the adenyl cyclase. This adenyl cyclase is it leads to cyclic AMP. Now what the cyclic AMP does? Cyclic AMP opens up the ion channels, whether the sodium or a calcium ion channel, and that further enhances the synaptic activity. That's one. Or a guanylyl cyclase. So this guanylyl cyclase, again the same thing as cyclic AMP. So now in case of the G protein coupled receptors, so we have the alpha gamma units of the G protein. So the alpha gamma units, the alpha units activate the diacyl glycerol, gamma units activate the inositol triphosphate. Now, our gamma units directly link with the adenyl cyclase, cyclic AMP formation, or directly linked with cyclic AMP formation, that they will directly open up the ion channels. So that means we have these ligands through the IP3 mechanisms opens the calcium channels, or they activate the calcium induced calcium release. That means calcium activating a calmodulin, so that calmodulin releases calcium from the calcium store, that is the endoplasmic reticulum. In case of the skeletal muscles, it is sarcoplasmic reticulum. And diacetyl glycerol, that activation of diacetyl glycerol activates the membrane bound phospholipase C and phospholipase A activity. And this phospholipase C again that will open up the ion channel there, or phospholipase A that will bring about prostaglandin or prostacyclin changes. Then I have mentioned about metabotropic actions. The metabotropic actions, because sitting on the ligands, they would bring about the nuclear changes for the growth of the neurons, or the proliferation of the neurons, or the synaptogenesis of the neurons, or dendritic arborization of the neurons. These are the metabotropic changes that is brought about by the nuclear changes in the neurons. These are in relation to the long-term changes. So whenever I am doing certain actions, so these actions, if I keep repeating, these metabotropic actions become an important thing in terms of our memory, learning, and our experiences. So these are the signaling pathways in broad about a neurotransmitter action. So now a brief about the excitotoxicity. Somewhere back, I just try to, I was mentioning about the excitotoxicity. Here I am just reading these lines. The most of the excitatory transmitters. Here I have taken an example of a glutamate. Usually the glutamate is cleared by the synaptic part by the reuptake mechanism. And this reuptake mechanism is a sodium dependent reuptake mechanism. That means there's a glutamate transporters in the neurons, presynaptic terminal, and on the glia, because they have a glia by the side. So thus they minimize or the lower the level of glutamate at the synaptic place. If it is high in this, usually in the chemical in the extracellular fluid space or inside the neuron, it is in millimolar concentration. So here we are talking about it minimizes it to micromolar levels and millimolar levels. This is 1000 times. The micro to milli is 1000 times. So that means they will, this is 1000 times lesser here. 1000 times lesser is, if it is coming up to millimolar level, that would definitely produce certain effects. So these excessive levels, what they are triggered, they occur when there is a ischemia, anoxia, hypoglycemia, or trauma. So when that happens, because of the excess, they open up the calcium channels. There is an enormous amount of calcium enters in. The calcium entry triggers the number of cellular mechanisms. All the lysosomes or lysozymes, they are activated. The release of those lytic enzymes that would kill the neurons. So that means the neurons die. That means they produce the death of this. This is excitotoxicity. Okay, so that means any excitatory neurotransmitter in excess that would kill the neuron by itself because it cannot stand the calcium influx. Now, what happens in case of ischemia? So whenever cerebral artery is occluded, say as happens in case of a stroke, there are, what happens in the area where the artery supply, the central portion, there is a ischemic area. Definitely that ischemic area will die. But the surround area, the ischemic area and surround, that surround area is known as a penumbra. The surround area is also partially ischemic, and that may survive. But what happens? They lose their ability to maintain the sodium gradient, so especially the functions. So the polarized state of the membrane is lost. So that means the resting membrane potential is not maintained. The elevated intracellular sodium, that means a depolarized state, this prevent the ability of astrocyte to remove it. So that means it happens to all cells. The astrocytes will not be able to take up the glutamate. So in turn, activate the excitotoxicity and death. And this happens in the surround area. So that means we have an area which is divided because there is a stroke. There is a part of the artery supplying that area. That central part is gone. The surrounding part is also going because of the sodium. There is excess of sodium and a depolarization. And this could be, suppose if this we can prevent, that would be our aim whenever we are having a stroke or a stroke affected individuals. So this is about excitotoxicity. Now, so I stop here. I have mentioned in this lecture, I have mentioned about the vesicular associated membrane proteins, how they work, how they move, and various concepts regarding that. The second, I have mentioned about the neurotransmitters, the various neurotransmitters, and their excitatory neurotransmitters. I have mentioned about, or I have listed about the inhibitory neurotransmitters, and then I have talked about their how they produce the action. What I have not gone into the details of each of these neurotransmitters. I would love you should go to the genome, wherein one full chapter is dedicated for neurotransmitters. I want you to read. That's a simple chapter which can be easily which is easily understandable. Everybody should read that. Then we come back. I will not be teaching that particular neurotransmitter chapter in the entire. You can go to the genome and read that, and you will have a good detailed aspects about the neurotransmission. Different neurotransmitters are explained in a very simplified manner. Okay, so now with this thing, I had given other day your class representative asked me what to do with or when to submit the assignment. So now I have written it very clearly to be written in your assignment book. So there are certain things I want that you write in your book. To write only 2 to 3 at the maximum 5 sentences on each of these, so that would enhance your knowledge about the particular chapter or particular topic. Now here I read, list the various events in vesicular recycling with the help of a diagram. I have given you the diagram. Now you mentioned about what is docking system. Okay, then you think about what is a kiss and run event, that is the exocytosis. Then write short notes on synaptobrevin and syntaxin. One is a vesicle associated protein, one is a membrane associated protein. You mentioned about that, how they are working. Or there may be a short note on vesicle associated membrane proteins. So these are also known as synaptogamins. These are known as. So that means you may get a short note, something like that. So now, what is zipping mechanism or unzipping mechanism? Just I have mentioned in my diagram. So then, what are the various requirements for a neurotransmitters? I listed. Then we have a neurotransmitter and receptor interaction. I have dealt with these aspects. Then I have differentiated between the botulinum and tetanus toxins in greater detail, because this is a question which often comes for your examination. Then I briefly mentioned about excitotoxicity. This is not enough. Excitotoxicity is a very big chapter. So, but just briefly introduced you to excitotoxicity. So then I would love that list excitatory and inhibitory neurotransmitters. At least your aim is, without thinking, try to write 5 each in each of these items. Okay, this is about the assignment for today. Write in your assignment book. When you come back here after the opening, we will try to see it. And it's the only one thing what I say, that you are working for yourself. You have to be motivated to write. So rather than we check and do, and you can go back whether what you have written, there is what is called a self-assessment. You assess yourself whether you are able to do it or not. You are the best judge for yourself, because here you write it, but what I advise, you write it in points, so that you develop the habit of writing. Then these are the reference books. I would not miss them. The Candles Neuroscience book, Guidance Textbook of Medical Physiology, Genong's Review of Medical Physiology, and Samson writes. These are the books wherein I have made the concepts about the synaptic transmission. So the next class, what I would be dealing with the properties of the synapse, because again,